It turns out that S bacterial cells have a coating that prevents the mouse's immune system from recognizing the invader. The R cells have no such coating and are therefore readily attacked by the mouse's immune defenses. Through his involvement with public health, Griffith knew that multiple strains had sometimes been isolated from a single patient, and so he was curious about how different strains might interact in his unfortunate mice. With one combination, he made a remarkable discovery. When he injected heat-killed S bacteria, harmless, and normal R bacteria, also harmless, the mouse died. How could two harmless forms of bacteria conspire to become lethal? The clue came when he isolated the pneumococcus bacteria retrieved from the dead mice and discovered living S bacteria. It appeared the living, innocuous R bacteria had acquired something from the dead S variant. Whatever it was, that something had allowed the R, in the presence of the heat-killed S bacteria, to transform itself into a living killer S strain. Griffith confirmed that this change was for real by culturing the S bacteria from the dead mouse over several generations. The bacteria bred true for the S type, just as any regular S strain would. A genetic change had indeed occurred to the R bacteria injected into the mouse. Though this transformation phenomenon seemed to defy all understanding, Griffith's observations at first created little stir in the scientific world. Well, this was partly because Griffith was intensely private and so averse to large gatherings that he seldom attended scientific conferences. Once he had to be virtually forced to give a lecture. Bundled into a taxi and escorted to the hall by colleagues, he discoursed in a mumbled monotone, emphasizing an obscure corner of his microbiological work, but making no mention of bacterial transformation. Luckily, however, not everyone overlooked Griffith's breakthrough. Oswald Avery was also interested in the sugar-like coats of the pneumococcus. He set out to duplicate Griffith's experiment in order to isolate and characterize whatever it was that had caused those R cells to change to the S type. In 1944, Avery, MacLeod, and McCarty published their results, an exquisite set of experiments showing unequivocally that DNA was the transforming principle. Culturing the bacteria in the test tube rather than in mice made it much easier to search for the chemical identity of the transforming factor in the heat-killed S cells. Methodically destroying one by one the biochemical components of the heat-treated S cells, Avery and his group looked to see whether transformation was prevented. First, they degraded the sugar-like coat of the S bacteria. Transformation still occurred. The coat was not the transforming principle. Next, they used a mixture of two protein-destroying enzymes, trypsin and chymotrypsin, to degrade virtually all the proteins in the S cells. To their surprise, transformation was again unaffected. Next, they tried an enzyme, RNase, that breaks down RNA, ribonucleic acid, a second class of nucleic acid similar to DNA and possibly involved in protein synthesis. Again, transformation occurred. Finally, they came to DNA, exposing the S bacterial extracts to the DNA-destroying enzyme DNase. This time they hit a home run. All S-inducing activity ceased completely. The transforming factor was DNA. In part because of its bombshell implications, the resulting February 1944 paper by Avery, MacLeod, and McCarty met with a mixed response. Many geneticists accepted their conclusions. After all, DNA was found on every chromosome. Why shouldn't it be the genetic material? By contrast, however, most biochemists expressed doubt that DNA was a complex enough molecule to act as the repository of such a vast quantity of biological information. they continued to believe that proteins, the other component of chromosomes, would prove to be the hereditary substance. In principle, as the biochemists rightly noted, it would be much easier to encode a vast body of complex information using the 20-letter amino acid alphabet of proteins than the 4-letter nucleotide alphabet of DNA. Particularly vitriolic in his rejection of DNA as the genetic substance was Avery's own colleague at the Rockefeller Institute, the protein chemist Alfred Mirsky. By then, however, Avery was no longer scientifically active. The Rockefeller Institute had mandatorily retired him at age 65. Avery missed out on more than the opportunity to defend his work against the attacks of his colleagues. He was never awarded the Nobel Prize, which was certainly his due for identifying DNA as the transforming principle. Because the Nobel Committee makes its records public 50 years following each award, we now know that Avery's candidacy was blocked by the Swedish physical chemist Einar Hammarsten. Though Hammarsten's reputation was based largely on his having produced DNA samples of unprecedented high quality, he still believed genes to be an undiscovered class of proteins. In fact, even after the double helix was found, Hammarsten continued to insist that Avery should not receive the prize until after the mechanism of DNA transformation had been completely worked out. Avery died in 1955. Had he lived only a few more years, he would almost certainly have gotten the prize. When I arrived at Indiana University in the fall of 1947, with plans to pursue the gene for my PhD thesis, Avery's paper came up over and over in conversations. By then, no one doubted the reproducibility of his results, and more recent work coming out of the Rockefeller Institute made it all the less likely that proteins would prove to be the genetic actors in bacterial transformation. DNA had, at last, become an important objective for chemists setting their sights on the next breakthrough. In Cambridge, England, the canny Scottish chemist Alexander Todd rose to the challenge of identifying the chemical bonds that linked together nucleotides and DNA. By early 1951, his lab had proved that these links were always the same, such that the backbone of the DNA molecule was very regular. During the same period, the Austrian-born refugee Erwin Kargath at the College of Physicians and Surgeons of Columbia University used the new technique of paper chromatography to measure the relative amounts of the 4 DNA bases in DNA samples extracted from a variety of vertebrates and bacteria. While some species had DNA in which adenine and thymine predominated, others had DNA with more guanine and cytosine. The possibility thus presented itself that no two DNA molecules had the